Sure. So molecular docking is a computational tool that allows us to predict how two molecules, in our case, small organic molecule, that I will refer to as ligand hereafter, and protein binding site or protein pocket bind together, determining their preferred orientation. Molecular docking is very important in drug discovery projects because it helps us to understand how small molecules, which are potential drugs, bind to large target molecules, predominantly proteins or protein complexes, and help researchers identify potential drug candidates and optimize their structure. Machine learning has recently accelerated drug discovery process, analyzing vast biological and chemical data sets by training on those data sets to identify potential drug targets, design new drug compounds, and predict properties of those compounds. And in this work, I want to introduce machine learning-based molecular docking technique that we call RTDock, which is designed to increase accuracy of results in early stage drug discovery. And we perform several evaluations against widely used classical docking tools. In this slide, you can see a general scheme of RTDock architecture and inference pipeline. The tool consists of two parts. First part is artificial neural network that uses molecular graph representation of protein binding pocket and molecular graph representation of random conformer any given ligand. These two entities are processed by special type of artificial neural networks called graph neural networks and combined in intermolecular representation that contains information about both entities. And this intermolecular representation subsequently passed through intermolecular transformer neural network. We call it transformer because of similarity to well-known today large language models that also use transformer architecture under the hood. The result of neural network is protein ligand distance matrix with dimensions equal to number of atoms in protein pocket by number of atoms in ligand. And then we apply several fast algorithms that help us to convert this predicted distance matrix into some final ligand position. At first stage, we use gradient descent algorithm to get some point cloud approximate atom positions for the final ligand pose, assuming that all the atoms in input protein pocket structure are fixed in the space. And then we align input ligand conformer on this point cloud by changing its position, rotation, and changing torsional angles of the ligand to fit to this point cloud as close as possible. We use differential evolution algorithm for that. And in the end, we have final ligand pose relative to input protein pocket that we can visualize inside this pocket and understand how different atoms and groups of both molecules interact. For benchmarking, we used the recently released Blinder dataset that contains experimental data from protein data bank. And it is focused on protein ligand complexes that are experimentally resolved. probably the most significant advantage of this data set is its train test splitting process. Basically, how we split data into training subsets that will be used directly for model training and test subsets that will be used for final model evaluation. And we need to make sure that data between these sets is not intersected, and we can properly evaluate the model. So the blinder data set contains groups all these experimental structures in clusters by similarity. And by using this cluster property to divide the data into training and test splits, we can ensure that there is no protein ligand complexes in a test data set that have some noticeable similarity to training data. And we evaluate our model on the data that model haven't seen during training. And another useful property of this cluster-based approach is we can reduce test set redundancy. And we can do that by selecting one representative protein ligand complex from each cluster. And that way we can make sure that testing data contains representative non-duplicated entries and avoid over-representation of some popular drug targets or drug-like molecules in test data. So in the end, we assessed our tool on blinder test set and compared results to classical docking solutions such as Autodoc, Vina, and Glide. For evaluation and comparison, we used two accuracy metrics and one quality metrics. First accuracy matrix is canonical root mean square deviation, RMSD, between predicted and experimentally determined ligand pose. You can see formula in the slide in which delta represents distance between some atom I in predicted and reference structure. Another accuracy estimate is local distance difference test for protein ligand interactions, LDDT ELI. And generally speaking, it represents a fraction of interatomic contacts in predicted protein ligand complex that are correctly reproduced, since we know which contacts we expect from experimental structure. And the last estimate is Postbusters quality checks to assess physical and chemical correctness of predicted ligand poses. Many machine learning tools has issues with quality of its predictions, and these Postbusters quality checks let us understand if our predictions have correct covalent lengths, correct angles between covalent bonds, if our complex doesn't contain intramolecular or intermolecular steric clashes, etc. Okay, so let's now move to the results. This slide depicts performance on different docking tools, in particular machine learning based tool and classical tools. We compared to Autodoc 4 or Autodoc CPU, as we call it here, Autodoc GPU, Vina, and Glide, classical widely used docking tools. And these results depicts performance on blinder test set It's consisting of 675 distinct protein ligand complexes. And the machine learning-based tool is able to outperform classical tools in terms of root mean square deviation of atomic coordinates between predicted and experimental data. Also in terms of fraction of correctly reproduced interatomic contacts. We had some issues with quality of predictions. So we only had 72% of predicted ligand poses that pass all postbusters quality checks. And it mostly concerns issues with protein ligand steric clashes in predicted complexes. But we can apply fast post-processing local minimization using BINA tool or using universal force field and increase this fraction of predictions with sufficient quality to the levels of classical tools. Here is the distribution of accuracy metrics for the same test data. Classical tools tend to generate some noticeable fraction of predictions with high RMSD, high deviation between prediction and reference, and with low fraction of reproduced interatomic contacts. At the same time, machine learning-based tool is able to concentrate its prediction in zone with lower RMSD and high fraction of reproduced contacts. Previous test scenario represented so-called redocking case, in which we have experimentally determined protein ligand complex. We take ligand out of the complex, randomly change its conformation and position, and then redock it back into the protein pocket, which means that pocket conformation is already optimal, ideal for binding this particular ligand. And this is not what we deal with in real-life drug discovery projects, because we never know what is the optimal protein conformation, and we usually take conformation from different experimental structure, unbound from ligand or bound to different ligand, or we may use machine learning predicted protein conformations by machine learning, or we can sample some frames from molecular dynamic simulations. But in the end of the day, we don't know which conformation is optimal. And this test subset called Blinder MLSB is addressed to assess performance of docking tools in such ligand unbound scenario when we don't know optimal protein conformation. And this test subset consists of 344 protein ligand complexes. And instead of original protein structures, we replace original protein structures by experimental data from unbound from ligand, from protein conformations unbound from ligand, or we use predicted conformations by machine learning. So pocket structure is suboptimal for ligand binding. And you can see that compared to redocking scenario, accuracy of all the tools we compare dropped significantly. Still, machine learning-based tool is able to provide better results compared to classical tools in terms of both accuracy estimates. Again, we had issues with physical and chemical quality of predictions because of protein ligand steric clashes, but fast minimization led us to reach levels of quality similar to classical tools and retain superior performance in terms of accuracy. And this is distribution of accuracy metrics for the same data set. Here, most classical tools predict ligand poses with high deviation from experimental data and low fraction of reproduced interatomic contacts. And machine learning-based tool RTDoc still able to produce some noticeable fraction of predictions with good accuracy. And the last evaluation scenario is concentrated on heteroatoms. So heteroatoms is molecular entities in protein pockets, non-protein entities in protein pockets that might be important for protein ligand interactions, that they may include such molecules as such types of molecules as cofactors, ions, and the water molecules that mediate protein ligand interaction. So we divided original blinder test data into subsets according to presence of each type of heteroatom. And in all of these subsets, machine learning-based tool is able to outperform classical tools by both accuracy estimates. In conclusion, machine learning-based tool RTDoc predicts ligand poses more accurately compared to classical docking tools. It achieves top performance when optimal protein structure is unknown, and we use ligand-unbound protein conformation. And it is also more accurate than classical techniques in scenarios when protein binding pocket contains heteroatoms such as cofactors, ions, and water molecules. Thank you for your attention.
Thank you, Taras, for your interesting and important report. So, можливо, є запитання, як мають українському запитати? Немає питань?
Please tell me, I know I've been working with Autodoc Lina and Autodoc for the past couple of years, and I know that this tool is mostly used for non-covalent docking. This approach allows to find results of covalent and non-covalent docking. Yeah?
Only non-covalent.
Only non-covalent. Thank you.
Last question. So, RTDoc, it is not only software capable of prediction of molecular docking, where you pay attention to some other software, and what is the position of your developed software in comparison, for example, RTDoc or other software where machine learning is used? Are you familiar with other software?
Machine learning.
Okay. There are some of them software which were not using machine learning, but now they use, because they are trying to integrate machine learning in their classical approaches.
Yeah.
Did you compare? Did you do some analysis comparison of your software and software of other groups?
Yes, actually, this publication actually contains a wider comparison to a wider range of tools, including other machine learning-based alternatives, including AlphaFold 3 and its alternatives of cofolding rules. And I'm working on making this evaluation scope even wider right now. But the general idea of this tool is to provide something that is more accurate than classical approaches and most machine learning-based approaches, but also that also provides speed similar or better than all other tools. So basically, it's designed for high throughput virtual screening, where we need to screen millions, tens of millions, times billions of small organic molecules against some drug target to find most promising candidates. And of course, apart from accuracy, we critically need speed of the tools to make it cost efficient. And I didn't present all that in this presentation because it will take too much time, but essentially this tool, it provides accuracy higher than better than classical tools on the same level as best machine learning tools out there, and it's faster than most of machine learning tools and classical tools.
Thank you, Taras.
So it's much better. Some everything was already existing.
For it's a good choice for high throughput visual screening scenario. I can say it's much better.
Maybe, but you have not participated in any competition. No, there is some competition like task in the United States.
Yeah, of course. It takes several years to go through this process. Yes, that's true. According to these internal evaluations and comparisons to traditional tools, can outperform traditional tools. It's faster than machine learning based tools, so it can predict, let's say, position pose of one ligand in less than a second on average rate hardware. And if we compare it to some cutting edge, very accurate machine learning tools like AlphaFold 3, Boltz 2, they may take tens of seconds or minutes, so it's different magnitude of speed at the similar accuracy.
We have one more question.
First of all, thank you for the report. That was great. Wisdom, this new approach based on neural networks. And actually, about the last statement you said, you mentioned AlphaFold, the last version of AlphaFold, because it allows to perform docking quite precisely. And have you compared the results, or what is advantages of your approach in comparison with AlphaFold, except time consuming or effectiveness or efficiency?
So AlphaFold is a co-folding method that, apart from predicting ligand pose, also needs to predict full protein structure, and it takes, as I said, a lot of time, and sometimes minutes, sometimes tens of minutes, and in this particular publication, we had some comparison to co-folding tools, including AlphaFold 3, and accuracy is on the same level, approximately. And I believe RTDoc provides a few percent higher accuracy on this. This is different kind of data set that called POSIX, also recently released, also that already contained evaluation for results for wide range of tools. And yes, but RTDoc, of course, have many limitations compared to AlphaFold 3, because AlphaFold is universal. You don't need to select protein conformation. You don't need to think about where is binding pocket on your protein. Just provide protein sequence, ligand, and you get docked structure in 1 or several minutes. RTDoc is rigid pocket specific docking. So we need some rigid protein structure. We need the location, precise location of binding pocket. And in this scenario, we can achieve much better speed for such projects where we need to iterate through many drug candidates.
So it's not a blind docking.
No, it's pocket specific docking.
Thank you, Taras. And no? You don't have a question. Okay, thank you so much. And I want to invite our next speaker, Mikita Bogrilov.